Phytocure is the first precision medicine offering for dogs with cancer. For the last 30 years, we've known in humans and actually dogs and most animals that cancer is actually a genetic disease. It doesn't just occur spontaneously like we used to think. It actually occurs because of all these genetic abnormalities in the DNA of an animal. And when they accumulate to the point where the cells start dividing uncontrollably, that's just what we call cancer. So in humans, we've actually run sequencing to identify where in their 3.2 billion base pairs of DNA the mutations are. So what we've done as a company, we've taken the precision medicine approach that has been successful on the human side and adapted it for veterinary medicine.
Welcome to Dog Cancer Answers, where we help you help your dog with cancer. Here's your host, James Jacobson.
Hello, friend. Thank you for joining me this week on Dog Cancer Answers. This week's interview is long, but oh so very fascinating. You know, if you've ever Googled dog cancer, which I'm sure you may have at one point or another, otherwise you probably wouldn't be listening to my voice right now, in those search results, did you happen to notice that something called Phytocure stood out as the top of the search query? It's an ad, of course, a paid placement on Google, but my guess is that if you're anything like me with internet searches, you started heading down a rabbit hole, meaning you started Googling, what is Phytocure, or what does Phytocure do? And if you've listened to this show, Dog Cancer Answers, before, you probably already know that one of our missions is to proactively answer those kinds of dog cancer questions and get answers direct from the source. So sometimes those questions and answers come in the form of actionable veterinary advice or tips, full spectrum treatment options, and one of my favorite topics, which is introducing new and cutting edge cancer therapies. Therapies that will hopefully one day change the grim statistics of how many dogs cross over the Rainbow Bridge each year due to cancer. Which brings me to this week's guest, CSO of the One Health company, Ben Lewis. Ben is also one of the founders of Phytocure, and between him and his team, they have taken a groundbreaking approach to diagnosing and treating cancer in dogs using DNA sequencing. It's exciting stuff, right? The whole idea behind taking DNA, scanning it for cancer-causing mutations, and then creating a targeted therapy for that particular mutation almost sounds like something straight out of a sci-fi film. Well, it's actually pretty common first-line treatment protocol among many human cancers these days. And today, we are going to talk about how it has finally made its debut to the veterinary oncology space. From his Silicon Valley garage in California, literally, let's get started with this week's conversation with Ben Lewis.
I got kicked out of the main office, so I'm out here in the garage, but it works fine, hopefully.
I saw that. I was like, you're in a garage. Is this the legendary Silicon Valley garage?
So it's really funny. When we started out, we were a couple blocks away from the original HP garage, which was the start of Silicon Valley. But now we're, I guess, 4 or 5 blocks away.
Okay. As we record this, you are literally in a garage. I can see your garage door behind me and you're on the inside. So that's great.
This is a Silicon Valley garage. Yes.
I think that's actually a really good starting point to get into it, because we talked to so many different people on Dog Cancer Answers, from veterinary oncologists and all sorts of people in the space of taking care of dogs with cancer. But you're approaching it from a whole different One Health perspective, backed by deep pockets of Silicon Valley, right?
Mm-hmm.
So tell me about that.
So, as you said, you've spoken to, some of the great veterinary oncologists out there, and we're really taking a technology and kind of big data approach to cancer, just like has been done in the human space. So rather than basing decisions off looking at 3 or 4 or 5 dogs in prospective, randomized controlled clinical trials, we're doing kind of the new way of looking at data called real-world data and real-world evidence, which was really pioneered by people like Dr. Amy Abernathy, who actually used to be on the One Health team and is now the Deputy Commissioner of the FDA. It really looks at patients with real clinical problems with naturally occurring cancer that isn't exceptionally controlled. And it's more reflective of actual patients with the actual condition. If you're just going to look at cancer patients and you're going to cherry pick 4 or 5 of them, and all of them are young patients without any comorbidities that are otherwise perfectly healthy, that is generally not a reflection of most patients out there that veterinary oncologists are treating with cancer. They oftentimes have other things wrong with them. So at One Health Company, we've really taken the precision medicine approach that has been successful on the human side and adapted it for veterinary medicine.
Okay, well, why don't we start off by getting into it? What does Phytocure do?
So Phytocure is the first precision medicine offering for dogs with cancer. So, just like people, you diagnose a human being with cancer, but you don't just look at the type of cancer from a histopathological standpoint. For the last 30 years, we've known in humans and actually dogs and most animals that cancer is actually a genetic disease. It doesn't just occur spontaneously like we used to think. It actually occurs because of all these genetic abnormalities in the DNA of an animal. And when they accumulate to the point where the cells start dividing uncontrollably, that's just what we call cancer. So in humans, the first step after a histopathologic diagnosis, you actually run sequencing to identify where in their 3.2 billion base pairs of DNA the mutations are.
That's not done in all human cases, right?
For the most part, I think it's pretty well accepted.
Well, when you do genomic sequencing, but not everyone does that.
I think it's becoming pretty standard of care. I mean, for some like heme malignancies, for example, they might do something like flow cytometry, or they might look at neoantigens or neoepitopes if they're coming from an immunological standpoint. But I'd say sequencing is pretty standard for most cancers out there in humans.
Okay, but this is the first time it's being done with dogs.
This is the first time it's being done in dogs.
So how does it work? I mean, how do you get the sample? How does that work?
So we work with the veterinary oncology community. And just like they would diagnose a cancer histopathologically, they normally take a lump or a bump off. They send it to a veterinary pathologist. From there, the veterinary pathologist will just take a couple slices, sends it on to us. And usually around 3 weeks later, we can provide results back where we actually sequence a tumor using our next-gen sequencing panel, which is actually run in parallel with human samples. So literally, it's the same like CAP-CLIA certified lab that's running human samples, and we're actually running the doggy samples. And what's really cool is we actually get our results faster.
Why is that?
Because we do high volume and we have a great analysis pipeline from the bioinformatics standpoint. And we also have a great reporting team as well that actually generates the reports and sends them back to the veterinary oncologist with treatment recommendations.
So the actual analysis is done by your team, but it's actually being processed by the same facility that does human.
Correct.
Okay. That's pretty cool. But is it your team that's analyzing it, or is it like human doctors who are looking at it?
So it's our team that's actually analyzing it, but the sequencing is done on sequencers that are also running human samples.
Got it. Okay.
So Illumina HiSeq X10s, which are basically the most cutting edge gene sequencers out there.
Okay. And each of those sequencers costs a couple bucks.
A few dollars, yes. It's a pretty...
What does a sequencer cost? If one were to go on eBay and buy, a new one.
A couple $1 million.
Okay.
I think with the service packages, the X10s are around $10 million.
Okay.
Yeah. So they're expensive.
So that's why it's being done on the same systems that are being used for humans.
Correct.
So then it goes to your team.
Mm-hmm.
And is that composed of veterinarians or researchers or PhDs?
So our team is composed of PhDs, cancer biologists, researchers, bioinformatics, veterinary oncologists, human medical oncologists, pathologists. We have a pretty diverse team. And it really has taken a whole village to actually adapt this model from the human space into veterinary oncology and to do it in a way that, makes sense and is actually clinically accessible. So one of the big differences between human oncology and veterinary oncology, in human oncology, there's somewhere like 150 FDA approved drugs for people with cancer. In veterinary oncology, there's one.
That is specific to dogs.
To dogs, yeah.
Yeah, Palladia.
Palladia, yep. And that was developed by Dr. Jerry Post, who's our chief medical officer, and Dr. Cheryl London, who's on our scientific advisory board. And it's obviously sold and distributed by Zoetis.
Okay. And then so for all the, people wondering, but there's so many treatments that they have for dogs with cancer. Those are all human chemotherapy drugs.
Correct.
And you're saying that basically what Phytocure enables veterinarians to do is to more precisely use a human chemo drug on a dog. And I'm just trying to make sure that I'm clear on this, but oftentimes use human chemo drugs that most veterinarians would never even consider. because it's not standard of care for veterinary oncologists. True?
Yeah, you're like 60% of the way there.
Okay.
So most of the drugs that veterinary oncologists use today are chemotherapies, cytotoxic chemotherapies, which were kind of let's say the previous generation of cancer therapies in people. The current generation of cancer therapies in people are targeted therapies. So they're therapies that you use when you know what the underlying mutation is, and you try to specifically target just those cells with that mutation. And this is in kind of juxtaposition to cytotoxic chemotherapy, which kills basically any rapidly dividing cell, whether it's the bone marrow, whether it's the cancer, whether it's the lining of GI tract. That's what will be targeted by cytotoxic chemotherapy, whereas a targeted therapy specifically inhibits the growth of ideally just those cells with a given mutation.
Okay, so let's unpack that a little bit for people who are just overwhelmed because their dog has cancer.
Sorry.
So cytotoxic means, we'll just break that apart. That's toxic to cells. And that is the type of chemotherapy that is traditionally used with dog cancer. You're saying it's sort of an older type of chemotherapy. And in the human world, that has evolved so that while they still use some of those cytotoxic chemotherapy agents, there are a whole host of new drugs that are being used because of this genomic testing. Am I getting closer?
Yes, you're spot on now. So people have probably heard of BRCA1 mutations or HER2 mutations in breast cancer, BRAF mutations in melanoma, or EGFR mutations in lung cancer in humans. And it turns out dogs actually have these same mutations. And what's even more fascinating is the drugs that are being used in people today were originally tested in dogs. So we actually have a ton of data, particularly around safety and toxicology, about these drugs that's even older than their use in humans.
Right. Well, my understanding isn't like almost in all human drugs, the last species before it gets to human testing are dogs.
Yeah, so dogs and non-human primates are the last species before you can put something into. They're usually pre-IND, pre-investigational new drug.
Because human cancer and dog cancer is so similar.
Yes. It truly is remarkable. We see oftentimes, if you look at diffuse large B-cell lymphoma in people and in dogs, it's almost identical. Osteosarcoma. Angiosarcoma in people is the same thing as hemangiosarcoma in dogs. So this whole comparative oncology approach is amazing. And what we've done as a company, and as well as the whole ecosystem of academics and the government, NCI, National Cancer Institute, has done an incredible job of demonstrating is that at the genetic level, as well as the histopathologic and likely immunologic levels, canine cancer and human cancer is almost identical, which is really exciting because it does open up a whole toolbox that was previously unavailable. There was no reason before One Health Company and FidoCure came along to say, let's use this EGFR inhibitor in this type of doggy cancer, or let's use this BRAF inhibitor in this type of dog cancer, because we didn't know that mutation existed and in what percentage. So what we've done as kind of a company is we've really identified a whole variety of mutations. We had a poster at, I believe it was AACR, the American Association of Cancer Researchers, the triple meeting, where we demonstrated 64 mutations that kind of are concomitant between humans as well as canines. And a lot of these had never been demonstrated before. So it was like 1,000 dog data set, which is huge for the veterinary oncology space or comparative oncology space.
That's very large because many drugs are done in like 16 very unfortunate beagles.
Yes.
Okay, so let's unpack this again. So you got a dog with cancer, they take a biopsy, they send it to your mystery lab somewhere in, where is this?
It's in New Jersey.
Okay. So send it to New Jersey. It's sitting next to human. It's being sampled at the same time that human and other dogs. Are there other animals that are also processed through this same genetic testing?
We're just doing dogs and maybe a couple de novo cat sequences.
Okay. And then the data after the sequencing goes to your team of scientists, PhD researchers, who then do what?
Who then apply what are called drug rules to the data. So let's very simplistically break it down. It's almost like heuristics. If this, then not that. And people are really good at keeping track of, 1, 2, 3 of these and they're at the top of their head. But it turns out computers are incredible at actually keeping track of tens of thousands of them and coming up with, rank orders and whatnot. So, a lot of what Phytocure has developed over the past couple of years are these drug rules. It's kind of a real-time feedback loop. So they get better and better with each additional dog. So it's almost like a Bayesian statistic trial.
What's Bayesian statistic trial?
So think of it as like an adaptive trial, a system that has a feedback loop where each additional case improves the rules dramatically.
Couldn't we call that AI?
Yeah, you could call it AI, machine learning.
Machine learning.
It's a similar approach that Ray Dalio and Bridgewater Associates uses for trading stocks in their hedge fund. And it's the same approach that most other sectors have seen the application of AI, except veterinary oncology is still pretty far behind.
I get that totally. Okay, so are these drug rules that you originally started with all from the human space?
So a lot of them came from the human space, and then a lot of them were kind of extrapolated based on medical records. So, we have a whole team that all they do is...
Can you elaborate on that? What do you mean extrapolated based on medical records?
So it turns out taking the real world evidence approach where you actually structure data that was previously unstructured and take a look back. Okay, here's 500 dogs with hemangiosarcoma. They're all the same breed. These ones were treated with this. These ones were treated with this. Oh, the ones treated with drug X seem to live 40% longer than the ones treated with drug Y. You can then forward test that hypothesis and either prove it or disprove it. So there's just a lot of data that has always existed in veterinary medicine, but it hasn't really been mined very much. So that's one of the things we're kind of doing on the back end of PhytoCure is trying to identify these kind of latent data trends and drug rules that just, pop out when you actually start looking at the data and organizing it and analyzing it in large numbers.
Where are you getting the data?
So the data comes from a whole bunch of agreements, and a lot of it is actually coming from our PhytoCure patients.
Obviously, you can do it now with your PhytoCure patients, but before you were getting the data that you were combining with the human rules. And so I'm just wondering how much data how many years or like how many dogs?
So a lot of that actually came from papers originally. So a lot of it was coming from like the publications because that is kind of the strongest level of evidence that we could start with. And then as we've gathered more and more data from more and more patients, we've been able to refine those rules.
Was there enough? Because I would imagine those older papers were basically talking about the traditional cytotoxic chemotherapy agents.
Right.
So how did you extrapolate that to these more new modern class of drugs?
So not all patients actually have what are called actionable mutations. So in dogs, we see somewhere around 65 to maybe 67% of patients have what's called an actionable mutation, which means they have a DNA mutation that is actually druggable. Meaning the patient has a mutation that's targetable with a drug that we have access to.
So that means about 30-40% do not.
Correct. And in those cases, we can work with the oncologist to help identify what is a potential next step for that patient. And a lot of these patients are also treatment refractory, which complicates things.
Okay, well, let's again break down all these words for our medical listeners. What is non-refractory?
So there's treatment naive, which means a patient that's never seen a drug or radiation or any other, let's say, therapy. And then there's treatment refractory, which means they're a second-line patient or a third-line patient, which means they've already seen a round of therapy or several rounds of therapy and have failed. So that's kind of treating in a salvage in a different rule base than these patients that have never seen any drugs.
Okay, so going back to the 60-something% that could benefit from this, I'm assuming, just to pull that out, that means that about 60% of dog cancers might be appropriate for this type of treatment regimen, but you don't know until you do the testing?
Correct. So there's a great paper by Gwen Onwang and Nikki Mason over at University of Pennsylvania looking at hemangiosarcoma, which is a very common cancer of the lining of the blood vessels. And it's commonly seen on an emergent basis where the dog comes to the emergency room with a ruptured spleen. So there's 4 or 5 different subtypes of hemangiosarcoma, it turns out. And some of those subtypes, we theoretically have drugs that can slow the growth or the progression of that disease. And then there's other subtypes where there's no known drugs. So an example might be, let's say, if you have the P53 mutant version, there's no therapies that can, inhibit the growth of P53 mutant cells because P53 is what's called a tumor suppressor gene. Most of these actionable mutations are what we call proto-oncogenes. I can get into it or I can just punt the topic.
I think if our listeners are this far, we call them dog health geeks.
So an oncogene is basically the gene that when it's normal or what we call wild type, that gene basically is functioning normal. But when it's mutated, it then can lead to cancer, which is uncontrolled cell division. So if you have a mutation, it's like putting the gas on. That's an oncogene. The opposite of that is like the brake, and that's what we call a tumor suppressor gene. So P53, when it's working, it puts the brake on cell division. But when it's mutated, it's like the brakes come off. Mm-hmm. So for those tumor suppressor genes, they're oftentimes more difficult to drug at this point. Although there are some really cool things. If you really want to nerd out, I could go way, way, way deeper into this.
Give me an example of something nerdy and geeky that would be fun.
Okay. So in human medicine, I'd say in the last couple of years, there's been a huge breakthrough, and it's something called synthetic lethality.
I love the name.
Sounds like a good movie, right?
Yeah, it does sound like Arnold Schwarzenegger.
Yep. So a lot of this work, I believe, was actually one of our advisors, Dr. Elizabeth Irons. She's the CEO of a company called Scientific. I'm blanking on the name, but she's an incredible PhD cancer biologist.
We'll put a link in the show notes.
Yes, Science Exchange. But she was part of the team that helped develop a drug called olaparib. Olaparib is a PARP inhibitor. So BRCA1 and BRCA2 are two common mutations seen in women with breast cancer. And previously, those mutations were untreatable because if you had, a bad copy or two bad copies of that gene, I think in the tumor suppressor gene category, there was nothing you could do really to specifically inhibit BRCA. But they realized that if you have one bad gene, the cells can continue dividing. But for some reason, when you have two bad tumor suppressor genes, for some reason, it kicks the cells into realizing that there's some mutations and they should stop dividing. And they kind of puts them on the path of programmed cell death. So what it does is it almost like fakes like a second genetic mutation so that the cell stops dividing because it thinks it's damaged, which it is damaged because it obviously has cancer. But it's a really fascinating new approach. And hopefully we'll see a lot more drugs kind of taking advantage of this synthetic lethality pathway.
Cool. Okay. So you got the test, you got the results, you have matched it with some drugs that you think will work. Those are almost all oral agents. They're not injected, or is it a combination?
No. So that's one of the big things that we've kind of worked around. Most of the therapies we're using are what are called small molecule tyrosine kinase inhibitors, which are all oral capsules. And they're all human FDA approved cancer therapies that we've brought back into the veterinary space via one of our partners that's a compounding pharmacy. So we'll work with a vet. They'll get the report that says, try drug X, Y, or Z. Mm-hmm. They'll then contact the pharmacy, and then based on either a mgs per kilogram or milligrams per meter squared basis, we'll actually prescribe their patients the therapy. And, all the therapies within the formulary are oral capsules.
Westwood Compounding.
Wedgwood.
Yep. Sorry.
We're working with Wedgwood Compounding Pharmacy.
Okay. And so that's, again, sort of a differentiator because so many traditional chemotherapies are done via IV, which obviously during COVID is even more problematic.
Yes. So there's a reason why we're really leaning towards the oral therapies. One is during COVID, they're a lot easier to administer. They're administered at home by the pet parent. Two, their kind of duration of action, if you will, is a lot shorter. So with an IV chemotherapy, once it's in the vein, you can't take it out again. And then you have to wait 14 or sometimes even longer for the effects of that drug to run their course. The nice thing about these small molecules is they often have half-lives like 12, 24 hours. So God forbid there even was a side effect, which, when you're dealing with cancer drugs, there are side effects. Thankfully, this class of drugs is safer than cytotoxic chemotherapy, at least based on our early data. And there's a poster where Dr. Jerry Post presented these data. I believe it was at the Veterinary Cancer Society meeting or the American College of Veterinary Internal Medicine meeting a couple of months ago. It was an online meeting. And because they have such short half-lives, if you just stop giving the drug, theoretically the side effect should just go away. So that's one of the other beautiful aspects of these small molecule inhibitors. They're oral, they're given at home. They have short duration, which, God forbid there was a side effect, it would go away rather quickly, hopefully. And it's just ease of administration. You don't have to bring your dog in, every 2 weeks or every other week, 1 week for blood work, 1 week for administration of chemo, 1 week for blood work, whatever schedule you and your veterinary oncologist have. So it's a lot less intensive, especially during COVID, which is important.
Let's talk a little bit about safety and efficacy in general as compared to a traditional way of approaching this if you go the oncology route in the first place. So how would you gauge the safety? And then let's do efficacy.
So from the safety standpoint, veterinary drugs, there's a whole veterinary oncology grading scale. I can send it to you if that's interesting to anybody. And we keep pretty copious records on the patients, and we collect adverse event forms for basically every patient that has an adverse event. We have a whole committee that adjudicates adverse events. Did the dog die because it was run over by the car? Or did the dog die because it was on a cancer therapy? Or did the dog have, elevation of liver enzymes because of the cancer, because of the therapy? And we have a whole team that, adjudicates this stuff. We're very, conservative when it comes to classifying adverse events. So we probably have a lot of AEs in there that probably don't belong, but just for statistics and whatnot, we do keep them in. And from an adverse event standpoint, the drugs that we work with are all safer, less toxicity than we've seen with, let's say, Palladia as an example. I think that was the benchmark. And I don't want to make any label claims or drug claims here,
Right.
just to be clear.
Right.
This is just observational data from, I think the poster had 722 patients in it. So it's a very large data set. Thankfully, in this day of digital records, we can actually gather the records fairly simply. And it turns out veterinarians as well as pet parents are very eager to report adverse events, side effects as they happen, when they happen, and for as long as they happen.
Yeah, people are oftentimes more willing to share that about their dog than about human beings.
Absolutely. And we very much welcome it because this is how we learn. And the sooner we know, and the more details that the veterinarians, and the veterinary technicians, and the pet parents can provide us, the better.
Okay, so that's the safety. What about efficacy?
So Christina, one of the things she said is I can't make any efficacy claims because they have not been reviewed by the FDA. We have had, let's say, based on real world evidence and early data, I'm just going to say we have statistical significance on several cancer types that patients who undergo Phytocure therapy are living longer. And let's say in hemangiosarcoma, it's like 150% longer. So it's considerably longer, which is exciting. And we have data even based on the next level. So it turns out hemangiosarcoma is kind of a catch-all term. There's probably four, as alluding to the Guanan Wing paper on hemangio, there's 4 or 5 different types of hemangio. So it's basically 4 or 5 separate diseases that we just kind of group together as one based on what it looks like under a microscope, when we should be characterizing these things based on the mutation that causes them. So we have data down to the specific mutation. So if I started saying, which mutations have better and worse prognoses, I'm sure my team would kill me.
That's when the lawyers call you.
Yeah.
Yeah.
That's when Christina comes downstairs and starts yelling at me.
That's your wife, your co-founder.
That's my wife and my co-founder.
And your boss, yeah. She's the CEO.
My boss.
What's your title?
I'm the CSO, the Chief Strategy Officer.
And not to be confused with the Chief Scientific Officer. Okay, so she's the boss, and so she wouldn't have you getting the micro details of the efficacy. Okay, how many dogs have gone through Phytocure at this point?
At this point, I think we're around 1,200,
okay,
which is by far the biggest veterinary oncology data set in existence by at least an order of magnitude, which is exciting.
So when Dr. Post did Palladia, how many dogs did he work with?
I don't have the data, but if I recall correctly, it was on 94, 96 dogs. Mm-hmm. But I haven't read the FDA submission lately.
And the reason we're talking about Palladia is, as we said earlier, that was the first and only, actually there were 2, but it was the only one that's currently in existence today that is specifically designed for veterinary use.
I was reading a paper from basically the top veterinary oncologists at all these academic institutions looking at the efficacy of another veterinary oncology vaccine, and they had 8 patients. Another therapy, was put forward with 9 patients. Mm-hmm. So, at 1,200, we're kind of hitting new limits here. And it's really exciting because we're not just using novel therapies, but we're also looking at cancer in a whole new way based on the underlying mutation that's causing it.
Can you elaborate on that? What do you mean looking at cancer in a whole new way?
So based on the work of Dr. Bishop up at UCSF in like the late 90s, we realized that for the last 100 plus years, we've categorized cancer based on what it looks like under a microscope, because that's what we've had, what the cancer looks like and where it comes from. But Dr. Bishop won the Nobel Prize in like the late 90s for demonstrating that cancer is a genetic disease. So once you realize that cancer is a genetic disease, you realize, okay, so the cause of cancer are these genetic mutations. And if you can identify what the mutations are, you can theoretically extend life. And in humans, that's been proven. And that's what we're demonstrating in dogs as well. So we no longer look at breast cancer is just some type of lump in the breast. Is it BRCA1 positive, BRCA2 positive, HER2 positive? Let's say melanoma. Is it V600E BRAF positive, or is it EGFR positive and KRAS positive colon cancer? And there's two kind of components of that. One is the histopathologic diagnosis, which is what it looks like under a microscope, kind of the tissue of origin and where it is. And the equally important part is what's actually causing it. So think of cells like little computers, but instead of zeros and ones, computers are binary code.
Right.
Cells are quaternary code. So A's, C's, T's, and G's, like Gattaca. And the cellular programming is all contained in the DNA. So when you have a bug somewhere in the DNA or a mutation in the DNA, sometimes that puts the individual cells on these programs that just tells them to divide and divide and divide. And that's what we call cancer. So if you look at, doggy bladder cancer, transitional cell carcinoma, according to the work of Dr. Breen at North Carolina State University, somewhere around 85% of them are V5E, that's amino acid change, BRAF, which is the gene, positive, which means 85% of dogs have the same mutation for their bladder cancer. And why that's interesting is basically the vast majority of bladder cancers fit into the 85% bucket, and then 15% fit in the other bucket. But those are basically two separate diseases despite occurring in the same place. I guess a good analogy would be like if you went to the doctor right now with a cough. A cough is a cough, right? Your doctor would say, you've got a cough. But you probably wouldn't be satisfied with, you got a cough and this is what it sounds like. It's a loud cough, it's a quiet cough, it's a dry cough, it's a wet cough. But you probably want to know what's causing that cough, right? Like, do you have COVID? Do you have asthma? Or did you swallow, I don't know, did you aspirate a piece of pasta?
Mm-hmm.
Very different causes to the same physical manifestation of a symptom. And cancer is very much the same way. Understanding what the underlying mutation is actually gets to the actual cause of the cancer a lot better than just looking at the cells under a microscope and describing what they look like and where they come from.
Let's take a break right now, but when we get back, I'd like you to discuss the types of dog cancer that Phytocure works best with. We'll be right back after this important message. Welcome back. We are speaking with Ben Lewis, co-founder of Phytocure. So what kinds of cancer does this work best with?
So for the most part, I think the approach works really well with most types of cancer because, as cancer is cancer. And somewhere in there, 3.2 billion base pairs of DNA are the mutations. That being said, from the formulary we have access to currently, at Phytocure, we're really focusing on solid tumors. So we think lymphoma is interesting and leukemia is interesting, but I think that's kind of a place we've shied away from because the current standards of care are quite effective, particularly for like B-cell lymphoma.
Right. I mean, there's the CHOP protocol, which is probably one of the few things that pretty much makes a lot of sense.
Yes.
If you have a dog with that, it's like, let's use the Wisconsin CHOP protocol.
Yep, exactly.
But some of these other ones that you're talking about, like TCC, transitional cell carcinoma, and all these sarcomas, normally not so good. And that's what this is for.
Correct. So that's what we're really focusing on. Is, the hemangiosarcomas, the transitional cell carcinomas. Lung cancer is actually a great one. Primary pulmonary adenocarcinoma turns out to be very similar to what we see in humans. And melanoma, the list goes on and on, but basically solid tumors. And for us to sequence, we really do require a biopsy. Sometimes veterinarians will do what's called a fine needle aspirate, which is just taking a needle and kind of taking a small sample and squirting the cells onto a slide. But for sequencing, we really do require a chunk of the actual piece of the tumor. So either, a punch biopsy, a true cut biopsy, or just an excisional biopsy via surgery. So it's really important that we actually have some tissue. So that's another reason why we're focusing on the solid tumors.
Got it. Okay. Again, let's go back to the beginning. So you've heard about this. You've Googled dog cancer and you've seen one of Phytocure's millions of ads, and people are like, what is Phytocure? What is Phytocure? Which is partly why we're doing this conversation, because people are like, what is this? What is the process that you would go through? You'd go to the website, and then what happens next? How do you bring your vet into the picture?
The good news is we're working with a fair number of the veterinarian collegists in the United States already. I think the number is north of 30%.
So you have to work with an oncologist, not just a vet, but an oncologist.
So we very much prefer working with veterinary oncologists. There are case-to-case exceptions, just to be honest.
Because not every place, like I'm in Hawaii, where there are no veterinary oncologists here.
There's one veterinary oncologist, right, in all of Hawaii. Who is it? Is it Dr. McMillan?
On Honolulu, but not on Maui, I'll tell you. So, yeah.
Okay. So, if there's some reason why, a veterinary oncologist is completely out of the picture, there are possibilities for kind of exceptions. Let's say you are on Hawaii and you don't want to take your sick dog on an airplane to go receive chemotherapy. We really do want to help clients.
Right.
And that's our number one prerogative. So similarly, if you're in the middle of Montana or something, and there's literally not a veterinary oncologist in the entire state, we're very much, willing to work with on kind of a select basis, exceptional DVMs and VMDs who do like to treat cancer and are willing to kind of learn a bit. But we very much prefer veterinary oncologists. I'll double down on that.
And it requires someone at the beginning of the process who's a surgeon, because you really are going to need someone who can do the surgery part to make sure that you can actually get a sample.
Yeah. And for most of the tumor types that we're looking at, that's usually kind of the first step anyways, getting the histopathological diagnosis off of a piece of the tumor. So usually, dog has a lump or a bump, they go to their veterinarian. Vet says, that's looking suspicious. Let's take it out. Or the spleen has ruptured. Let's take it out in the middle of the night in the emergency room.
Right.
And then they'll send it to their reference pathology lab. Antech and IDEX are the two largest ones. And, if the veterinary oncologist would like to proceed at that point, they'll just have a piece of that tissue sent to us and we'll do the sequencing.
Okay. So that has to be done pretty much at the same time as it is sent to Antech, or no?
No. So actually, veterinary pathology labs have to keep the tissue for like 3 years. Depending on their state.
Okay.
So they actually save it. And we sequence off what's called formalin-fixed paraffin-embedded tissue, so FFPE tissue. So it could be 6 months later, a patient could have failed one round of chemotherapy and they're like, that first line therapy didn't work. Then the veterinary oncologist may recommend something like Phytocure, or they might recommend it as a first line. And while they're waiting for the sequencing results, they might start something else.
So that's good to know. So that sample is still at the pathology lab for 3 years. So later on, they come back and they say, it is transitional cell carcinoma, and this is the protocol. And then you say, hey, I want to try this Phytocure. Then your vet would order something that would basically get a sample from the Antech laboratory to your facility in New Jersey.
That's correct. Yep.
Okay.
And the client, the pet parent, doesn't have to worry about this stuff. This is all kind of done behind the scenes and actually quite straightforward.
Right. Okay. So this is all electronic, I imagine. And then you guys at Phytocure don't liaise at all with the dog owner, the dog lover, the dog parent. You are talking to primarily vet oncologists.
Correct. For the most part. I mean, plenty of pet parents do call us, and we have some incredible veterinary technicians, and I pick up the phone sometimes, Christina. So you never know who you're actually going to get.
Right.
And we love what we do, so that's why we still do it. But for the most part, we work behind the scenes with your veterinary oncologist.
Okay, cool. And so you're seeing good results at this point that are more promising than what you would see with traditional methodologies. Is that what you're saying?
Based off early data, the types of cancer that we're focusing on, I can preliminarily say yes.
Okay.
Hopefully that's like squishy enough that I don't get in trouble.
Okay. So what's the next step for you guys? Because I think what is interesting and where I want to go, because again, we're going to go back to the fact that you are sitting in a garage here in Silicon Valley, blocks away from where HP started, and you've raised, $16 million so far?
Sounds about right.
Okay. So where is this going? I mean, I like how it went. You changed your name from Phytocure and Ethical Pet Research, which I think is really interesting, right? To One Health Company. And One Health means something. What does it mean?
So One Health is kind of the commonality of diseases between people and animals. So, the most recent, let's say SARS-CoV variant 2, coronavirus, likely emanated from bats or something, or, some animal in some wet market somewhere. And that jumped the species barrier. So just from the disease standpoint, 75% of diseases that affect people are what we call zoonotic, which means they come from animals. So that's kind of a One Health interest. But One Health is really looking at the similarities between health and sickness between animals and people. And it turns out people and their pets are getting the same types of cancers, literally down to the genetic defect. So it's crazy. I mean, it's happened a few times where we've had a pet parent and their pet with the same type of cancer. And a couple of times they even have the same mutation. Maybe there's some environmental component. Who knows?
You think? We had the same for lawn chemicals or whatever. Wow. So again, help me understand where One Health fits into this, because again, that's a lot of money in the veterinary field. I'm sure, I don't have to tell you that. Clearly, there's something beyond just helping Fido, right?
Right. So what's really amazing is, because people and their pets are having the same cancer types with the same mutations, what we're learning from the dogs actually is going back and helping people too. So we worked with a large biopharma company on a study looking at human angiosarcoma, and based on actual dog data, they were able to see a signal for a label extension for one of their therapies in people. So that's really exciting. It's actually in, I think, phase 3 trials at Dana-Farber right now. So it's really cool to see, kind of this full circle of, it started in the dogs for testing, then went to people, then went back to dogs, and now is going back into people.
It's so cool. I remember reading a quote years ago from I think the director of the Dana-Farber Institute, who said that, like, if we can do it in dogs, we can do it in people, because they're so similar, these cancers.
And we've cured cancer many, many times over in rats, in rodent species. So it's not like curing cancer in a rat or a mouse, where we've done it many, many times. If you can do it in a dog, I think that's a much stronger signal.
Am I right in presuming that is why these large Silicon Valley venture firms have been interested in what you guys are doing?
So that it's part of it, this kind of comparative oncology aspect. But it turns out there's a lot of animal lovers out here in Silicon Valley, believe it or not, who have dogs who have suffered from cancer, and they themselves are cancer biologists. And when they go to their veterinary oncology clinic and they're offered a therapy from like the 1950s, their response is hell no. I know there's something better. Like, I want it. Please give it to me. I will pay anything. So realizing that didn't exist, and we were kind of in their backyard here in Silicon Valley, they decided to, help us kind of redefine veterinary oncology in an evidence-based way, as well as, expanding the reclassification of veterinary cancer and expanding the veterinary oncology toolbox with access to new therapies. So it was kind of a great merging. They love the veterinary aspect of actually helping dogs because they themselves are animal lovers. And if we can also help people, that's just kind of icing on the cake.
Well, what do you see as the future for the company as the chief strategy officer?
So the future of the company, I think in 10 years from now, Phytocure, or at least the Phytocure platform, will be the standard treatment methodology for most dogs with cancer. If you see a dog with cancer, your vet is just going to do Phytocure to kind of identify the right treatment, identify what's next, and go from there. So I think that's kind of the future. There's 6 million dogs diagnosed with cancer every year, unfortunately, and that's compared to about 1.6 million people.
And I think that's 6 million dogs in North America.
In North America, yes.
Yeah, you're not even touching the whole world. There are lots more dogs. So what does Phytocure cost?
So we leave the pricing up to the veterinary oncologists. And
give me a range, though, because we know that like a veterinarian in New York is going to charge more than a veterinarian in Ohio, probably.
Right.
So what's the range?
So I think that the most recent care credit data is the average cost of veterinary oncology care is $6,700 per dog, per incident. We aim to be considerably less than that. So one of the nice things about having these awesome Silicon Valley backers is they actually subsidize the cost considerably for pet parents.
That's an interesting angle.
Yeah. So, it turns out drugs from this century, as opposed to drugs from like the mid-1950s, 1960s, are a lot more expensive. And it turns out when you actually have to compound them on a named patient basis for each patient, that also adds costs instead of just, doing a run of 1 million pills in a given size. So what we do is expensive, and genetic sequencing is also really expensive. I'm just got a quote from a friend today. It was like $47,000 for whole genome sequencing in Italy, and with RNA-seq as well. So most people love their pets, but they're not going to pay $47,000.
Right.
Thankfully, the cost of this is nowhere near that, just because we run a lot of volume.
Is it the same type of test? Are you saying if you were to get the same test in Italy, it would be $47,000?
Potentially. It seemed really high to me, so I'm going to back away from that number. But in terms of sequencing in and of itself is expensive. And when we started, the cost of the test plus the lifetime of therapy literally was more than we paid for the cost of the test. And we were subsidizing it just because the data itself is interesting. And we really are a mission-based company that wants to help animals. So we've found other people that actually back this and support us helping animals, even if it means we lose money on every case.
Okay, so that's another value of the way you've structured your business, is that effectively because there's a bigger focus and a larger focus beyond dogs moving into the human space, the treatments are effectively being subsidized in order to get this data, which I imagine is something that people have to agree to as part of the process, that this is going to be data that's going to be shared and used in perpetuity. Now, on your website, it says there are 14 FDA human-approved drugs that you guys sort of use. Is that right?
Yeah, so that's kind of the initial tranche of therapies that we gained access to is 14 drugs, but going from 1 to 14 is pretty big. 14 compared to 150 is not that big. But, going from one targeted therapy in veterinary oncology and adding another 14, that is a significant increase.
Are the side effects that you're seeing from those 14 almost exactly the same as the side effects that you might see in human oncology with those 14 drugs?
Interestingly, no. So people and dogs can behave differently. Our shock organs are different, and our organs of toxicity can be different. And, if you talk to someone who focuses on allometric scaling or pharmacologist, sometimes we see similar side effects and sometimes we see different side effects or lack of side effects. So it has been a learning curve. But at this point where each drug that we use has been dosed in, hundreds of patients, we do feel pretty comfortable with the doses and the regimens that these patients are using.
Okay. But you're obviously, part of this whole discovery process, because this is still very much in its infancy in research, is to figure out a term often used in oncology is maximum tolerated dose. Do you guys focus on that at all with this or no?
So the whole MTD concept is really for cytotoxic chemotherapies.
Okay.
We're trying to kill the log maximum of cells, because just killing any cells it comes in contact with.
Right.
Whereas the targeted inhibitors, it's all about kind of area under the curve and making sure that you're actually inhibiting the inhibitory concentration, the right amount of cells for the right amount of time based on how often you're giving a drug, whether it's daily or whether it's every other day, making sure that you're inhibiting the growth of those cells with that mutation at that dose and that regimen.
Cool.
And that's completely different than gene therapy, and that's completely different than CAR T, chimeric antigen receptor T cell therapy or other cellular-based therapies, and completely different than things like, let's say, Listeria monocytogenes and other immuno-oncology vaccines. So we've seen it all because we do work with large human biopharma companies. And there's a lot of really cool stuff on the horizon that we're excited about. But today we're focusing on these 14 oral targeted therapies that have common mutations in dogs and we feel comfortable with.
Got it. And then in terms of the side effects, sometimes you see different side effects than you would see in a human. Are they sometimes worse or better, or how are they different?
So here's a good example. There's a drug, we don't use it, but there's a therapy called dabrafenib in humans. It's a BRAF inhibitor that's part of a doublet therapy for V600E BRAF positive melanoma in people. Turns out it's completely intolerable in dogs. So we never put it in a single dog because while being tolerable in people, it's completely intolerable in dogs. The corollary of that is maybe, let's say, I probably shouldn't name drugs that we actually do use, but let's say another drug that is totally tolerable in dogs might be less tolerable in people for some reason. And, might be because sometimes it's just idiosyncratic. We don't quite know every difference between humans and dogs as much as we wish we could. But at this point, we have a pretty well curated set of drugs that we do work with that we feel comfortable with. And I think we've demonstrated pretty well in these 700 something dogs are better tolerated than the therapies out there.
That is pretty cool. So I mentioned earlier that I want to learn a little bit more about you. You called it ethical animal research because?
So this is definitely a hot button. So the name was very, very unpopular, despite my initial insistence around the name, because I guess it implied that all other types of research that didn't use naturally occurring disease in pets was unethical.
Those poor beagles that I mentioned earlier.
Right. So, I have to keep my mouth shut on this one.
Okay.
But I have the utmost respect for our lab animal colleagues. And actually, one of our co-founders is the former president of the American College of Lab Animal Medicine, Dr. Ravi Talwani, is amazing. He's one of the leaders in laboratory animal medicine. And, the approach of rather than taking a healthy animal, inducing disease and then running tests on it, of actually using animals that have naturally occurring diseases that are likely closer to the disease you're trying to make artificially in an otherwise healthy animal probably is a much better animal model. So that's kind of the model we're promoting from a kind of a ethical standpoint. That being said, I don't want to cast stones.
Got it. Okay. There's an interesting story of how you and your wife met and how you started in all of this, because I'm very impressed at your ability to rattle off these scientific terms. So how did you get started?
So I was like a bioengineer undergrad. I went to vet school at Penn for 3.5 years, although I'm not a veterinarian.
3.5. Okay. So you almost had the VMD.
Almost had the VMD. I was very close.
Yeah.
And I hope to have it, someday soon. I haven't given up on it. But this, one health company has kind of taken me away. So I was a dual degree student at Penn. I was in the VMD MBA program. So it was the veterinary program and then MBA in healthcare management with Wharton.
So they combined that together, or was that of your own choosing?
A little bit of both.
Okay. So you helped to create that program, I guess. Sounds like you were an early student.
Yeah, we kind of worked on the curriculum and inked quite a bit around that. One of my classmates, Dr. Molly Dominguez as well.
So what year was this?
This was 2008.
Okay.
So 12 years ago. And so what started as ethical animal research was actually a class project for healthcare entrepreneurship, which is HCMG 867, which is taught by Dr. Kurtzman and Jeff Lipson. And a lot of great companies have actually spun out of that class. So companies like Warby Parker, Volano Vascular, MedCrypt, I want to say Grand Rounds, but I could be wrong. So a lot of the really cool companies have come out of this one class in Wharton. And this company started as a class project. And within a few weeks, we ended up with some pretty awesome biopharma contracts that ended up taking me out of school. So I didn't go back and finish the last couple months of clinical rotations at Penn Vet. So unfortunately, I'm not a veterinarian yet. Someday, hopefully.
Okay. So how did you and your wife meet, and how did you guys start building this company?
So I was in Brazil for spring break. I guess I'm very fond of taking sabbaticals from school and doing really cool things. So the first time I took a sabbatical from school was in undergrad at Dartmouth. So I realized I want to train for the Olympics. And my mom...
In what sport?
So I love kayaking. I'm from Miami, Florida. I've always grown up on the water and loved to paddle. Mm-hmm. And Dartmouth was a great school for that. I guess this would have been 2002. I wanted to train for the 2004 Olympics and decided to take a leave of absence. My mother said, "Oy vey, my stupid son, what's wrong with him?" And there's actually a really funny story. Why would he leave Dartmouth and go to train for the Olympics? I should have raised him better. And this is actually at a second night Seder. And another one of the parents, they were in like Westchester, New York. Another one of the parents says, "Oy, my stupid son just dropped out of Harvard and he moved to California with his friends and he's starting some type of book face company." And it was Mark Zuckerberg. So totally too bad Jewish sons here.
My goodness. Must have been a very interesting Seder.
Very interesting Seder. But long story short, so first time that I took a leave of absence from school, I ended up making the Olympic team, represented the United States in the 2004 Olympics in Athens, Greece, made it to the semifinals. Really cool experience. And did go back and finish. Then from vet school, I took a leave of absence to go to do the MBA. Once I was in the MBA, I took a leave of absence when I went down to Brazil on spring break. It was supposed to be a 2-week spring break trip and ended up turning into a 4-year spring break.
Got to get out of Philadelphia for 4 weeks. Okay.
Yeah. So I ended up in Sao Paulo for about 4 years, where one of my learning teammates, Bernardo Arspide, and I started up a company called 4Vets down in Brazil, which is, I guess one of the big animal health supply distribution companies in South America. And then we were acquired by a private equity fund, came back, went back to Wharton, took this class and was supposed to go back after one health company, but one health company is still going. So hopefully soon I'll go back and finish, whether it's at Penn or somewhere else, and, complete my VMD or DVM. But I met Christina in Brazil. So she was down there. She was managing director of Cerberus Capital, which is a large private equity fund. One day, one of our mutual friends from Germany brought her we kind of shared an office. He was with like a German private equity fund, brought her into the office. I spoke to Christina for about 5 minutes. I said, "Oh my God," looking over my business partner, "Oh my God, this girl's amazing. I'm going to marry her." And 6 months later, we were married. And after our second child was born, Christina was ready to kind of get back into the business world. And she saw what I was doing and she saw all the mistakes I was making. And she said, "Let me help you. You stick to the science, I'll stick to the business. And I think we can build something pretty amazing." And that was almost 4 years ago.
Wow. And then did you start in California or did you start somewhere else?
So we actually started in Philadelphia. So our original office was in the Science Center, I think, of University of Pennsylvania. It's kind of like tech offices right next door to Penn. and Drexel. So we started in Philly, and we originally moved to California for Y Combinator, which is an amazing accelerator program.
And for people who don't know what that is, what is Y Combinator?
So Y Combinator is this just awesome accelerator program started by Paul Graham. It's based here in California, and you probably know a lot of the companies that have come out of this accelerator program, things like Dropbox, Airbnb, Instacart, Science Exchange, which I mentioned earlier, Dr. Elizabeth Irons, and just a whole laundry list. So we came out to Silicon Valley for Y Combinator and have basically been here ever since. So we're still here in basically downtown Palo Alto. The weather's amazing, although you're in Hawaii, so I think it might be a little nicer there.
You have a heavy coat on, but of course you're in the garage.
Right. I'm relegated to the garage. But yeah, California's been amazing. If you are looking to start a business, there are definitely some strategic reasons to be located here, even with COVID and a large exodus of some big companies from California. This really is a center of talent as well as capital in the United States. And it's just exciting living here because everyone around you is doing something cool.
So do you basically have a distributed team? How are you guys coping with COVID?
So we were distributed to begin with, long before COVID. One of the things Christina really focused on as a company is company culture. Mm-hmm. And, she wanted to build a very equitable company across kind of all lines, gender, cultural background. And, one of the things that can make it difficult to recruit highly competent and talented women is forcing them to move from one location to another, as well as sometimes the burdens of childcare and whatnot. So like Christina's supposed to be on this, but our nanny had to leave early and, couldn't make it. So one of the ways Christina really wanted to attract like top talent is with this flexibility. And this is before COVID. So it would have been unheard of to let someone work from Nebraska. If they're a great bioinformatics person and they want to work from New Jersey, that would have been anathema to a Silicon Valley company. But we really have had this distributed culture. We communicate via Slack and the whole G Suite. And we've been using Zoom long before Zoom became a COVID necessity.
Right.
So it's always been our company culture that people can work from wherever.
It's definitely the future way to build companies. And it's smart in so many ways.
And you're distributed as well, right? Fully distributed?
I've been distributed for many, many years, like 20 years.
That's amazing.
Yeah, because I really like Hawaii. And this is not a great place to be hiring a lot of people who want to work crazy hours. But what you guys are doing is really pretty cool. And I imagine as you look to the future, 5 years from now, what do you see?
5 years from now, hopefully we'll be like the Uber, the Lyft of veterinary cancer. Like if a dog has cancer, it's just automatically, you go through the Phytocure platform, basically synonymous with excellent veterinary care. And hopefully we expect this will remain, affordable in the veterinary oncology realm for hopefully forever. And hopefully we'll drive the price down even further. One of the things when we took money from Andreessen Horowitz, one of the conditions was this can't be a product just for the wealthiest 1% of Americans. This has to be something that's affordable. And, you have to do whatever to make it so that you actually address the needs of pet parents whose dogs have cancer. So hopefully we can continue driving down the cost to the point where people don't have to crack open their piggy bank too much to help their loved ones with cancer.
And I'm assuming the enabler behind all that is this data, which big pharma is going to use to help human cancer. We haven't said this explicitly, but I imagine that's your business plan. I imagine that's what they're interested in. So yeah, the dogs are great and we'll subsidize that. But what we're really after is this data so that we can get more information for human cancer.
Absolutely. And that's how we close the loop financially.
Well, Ben Lewis, thank you so much for being with us. We went way over, but I think this is really interesting and we'll make sure to share it with as many people as possible.
Awesome. Well, thank you so much.
Pretty fascinating stuff, huh? But what do real-world veterinarians think about Phytocure? Well, we asked two veterinarians who appear regularly on Dog Cancer Answers. The first is Dr. Nancy Reese.
So first, I have to say up front that I have not actually used Phytocure in any of my cancer patients. So all of my impressions are from researching the concepts behind the treatment. The idea behind Phytocure is that not all cancerous tumors, even tumors that are the same kind of cancer, not all those tumors will have the exact same genetic features. So for Phytocure, a biopsy of the tumor, either a piece of the tumor or the whole thing, can be sent in, and the tumor is then tested to find which genetic markers are on that particular individual's tumor. These genetic markers then help determine which treatments are more likely to have an effect on the tumor. We know, especially from a lot of studies in humans, that certain drugs will work better or worse depending on those genetic markers. And this makes sense in that we all know individuals who seem to react differently to medications than ourselves. That's likely due to a difference in how the body processes the drugs or how the drug affects someone because they have overall different genetics. For the tumors, we're looking not at the overall genetics of the dog, but of the genetics of that particular tumor. And this is such a great concept because it is an example of precision or personalized medicine. We are targeting that individual and tailoring treatments to that individual dog or human. And this is a different approach to treatment than just basing the treatment on the tissue of origin of the cancer. So theoretically, then, we would give medications that have a higher chance of having an effect on the cancer versus a blanketed treatment based on just the type of cancer. This would certainly be less wasteful in terms of not giving drugs that could have some toxic side effects, but we wouldn't be giving drugs that we know won't work on that particular dog's cancer. I think the company mentions 14 FDA human-approved drugs that they use, although I imagine that number would go up with time. And these medications could be given at home. And this is an obviously huge benefit, if effective for the dog, to be treated at home versus having to come into a clinic frequently. But for the less good side, this type of approach is still relatively new in dogs, so we don't have good studies that show the positive or the negative effects. We have some good anecdotal reports, but we really need some large, well-done studies to understand if this is an improvement of care. Another issue is that while this approach has long been done in humans, or at least longer than in dogs, the results are not as good as hoped. While there are, I think, something like 50 drugs used in humans to target the different genetic variations in the tumors, the cancers do not always respond to the targeted treatments. And when the cancer does respond, the response doesn't often last as long as we had hoped. These are not proven to cure the cancer, which would obviously be the best outcome. And while that can be said of conventional therapy, we are looking at things that are an improvement on conventional care. The other thing we really don't know is how this translates to the duration of effect in dogs. A lot needs to still be studied there. I think the potential for this approach is great. Precision medicine is certainly the wave of the future. But I think right now we're still in an information gathering phase, so I don't want to add any, quote, false hope for guardians. If conventional therapies are more effective, then I wouldn't want someone to skip a more effective treatment by going this route. But treating at home is a huge bonus. And again, the idea of treating the individual versus everyone the same is a great concept. I hope to someday say that this option is better approached than the current therapies, as we are all really hoping to improve the outcome for treating dogs with cancer. Thank you very much.
And here is Dr. Trina Hazza, a veterinary oncologist.
I love it. I love the idea of it. The difference, when you think of how it could be implemented, right? Phytocure is essentially a company that uses genomic markers or genetic markers to target specific mutations in cancer. So when you think of how we treat most animals that have cancer, for quite a long time, I've been doing this for over 10 years, we use majority chemotherapy, which essentially is what we call cytotoxic therapy. It targets the cancer cells themselves, and it creates death on the cancer cells through a variety of different mechanisms. But it also targets, when you think of cancer cells, you think that they divide very quickly. And so when you think of how high-dose chemotherapy works, it actually targets rapidly dividing cells. The problem with that is that, unfortunately, you get side effects with chemotherapy in about up to 30%, sometimes even more, depending on the chemotherapeutic you use. And that's because it doesn't just target the rapidly dividing cells, only the cancer cells. It also targets rapidly dividing cells that are throughout the body. And those are going to be cells of your GI tract or your bone marrow. And that's why when pets get chemotherapy, just like people, they can develop GI upset like vomiting and diarrhea or suppression of their bone marrow. Where with what they call targeted therapy, which is what Phytocure really supports, and that's really where their focus is they take the tumor that's sitting in the laboratory, your pet's tumor, they then evaluate it through a variety of different tests, and they look at the entire genome and they compare it to that and figure out where the mutation exists in your dog's tumor. right? Not just in your dog, because it's a whole different type of test, in your dog's tumor. So
just in the specific tumor.
Yeah. I think a lot of times people say, so if my dog has thyroid carcinoma, can't I just use this one therapy? And I'd say, yeah, and probably it'd work. However, it's not your dog's specific thyroid tumor. And that's what Phytocure does. It actually takes your dog's thyroid tumor, not just any dog's thyroid tumor. We'll take your dog's thyroid tumor, evaluate that specific tumor for genetic alterations. And based off of that, they implement what they call a targeted therapy. And so again, different than chemo that is more cytotoxic, kills the cancer cells, targeted therapy is considered what they call cytostatic. So it blocks the proliferation or the growth of the cancer. And so it looks at these specific molecular targets that are involved in the growth of the cancer, the progression, the spread of the cancer, and it actually blocks it. So what I tell owners when I try to explain the difference between targeted and chemotherapy is I say targeted therapy is almost like a light switch on, and that light switch is turning on the lights, and the lights are actually the cancer growing, right? It's like the electricity to tell the cancer to grow. If I have a drug that can go over to your light switch and turn it off, guess what happens? No more electricity to the lights or to the cancer, and it stops growing. That is the goal of targeted therapy.
So it doesn't kill it, but it stops it from growing and spreading and metastasizing.
And eventually, it can start to die off. We have tumors that are on targeted therapy that literally disappear, right? But it works differently than chemo that targets the cancer directly. This targets more, say, the growth factors and other things that are telling the cancer to grow and spread and so forth. So that's really kind of the philosophy behind Phytocure, and they believe in what they call personalized medicine. And that very much up my alley, because as an integrative oncologist, you have to practice personalized medicine. Every single dog that walks in, again, with thyroid cancer, let's say, isn't going to be the same dog and may not need the same type of alternative medication options or chemotherapy or targeted therapy. You have to look at the patient as a whole and decide what works best for that patient, not just what works best for that patient's cancer.
You've used Phytocure, right?
I have.
And you've had successes, failures, a little bit of both?
I would say I have absolutely had a little bit of both, just like with any modality, right? Anytime you're treating cancer, it's a difficult journey, right? I mean, there are certain tumors that respond and there's certain tumors that don't. I always think of this one case that I had with a very small little dog named Duchess that ended up having squamous cell carcinoma of the tonsil that had already spread to the regional lymph nodes. This is a very aggressive cancer. If you look at the numbers, the chance of a dog with tonsillar squamous cell carcinoma living 1 year is 0%. I mean, that's how aggressive this is. And I've been treating these tumors for, again, over 10 years being an oncologist, and I have never found a therapy that can work well enough. I can get a few months, I can try to get some pain-free time, but I have tried chemo, I have tried other modalities. I just cannot find the perfect combination of therapies or singular therapies that really work well. So I spoke to this particular owner. She was not interested in chemotherapy whatsoever and said, look, I would be interested in targeted therapy. This seems like the newer way to go, and I think it's absolutely true. This would be certainly a kind of a new wave of therapy in veterinary medicine and veterinary oncology. It's been around for a lot longer in human medicine, over 125 different targeted therapies in human medicine compared to the one that is FDA approved in veterinary medicine, which is insane. And so this owner said, look, Phytocure has, I think it's about 14 different therapeutic options that are FDA approved in people that they found can target some of the veterinary oncologic cancers and have been proven to be safe in non-tumor bearing dogs. And so we thought, well, what they did was they looked at what would be safety profile and said, why don't we try these doses and see? And they had been using it for a few years. And so I felt very comfortable having this conversation with the owner. She wanted to move forward in that direction. And we, of course, did some herbs and cannabinoid therapy along with Phytocure. The dog started to get a very sensitive stomach, so ended up stopping the herbs, continued with the cannabis and the Phytocure. And Duchess must have lived for, over 9 months and had a wonderful quality of life. Got to be home, getting the pills at home, because these are pills. So I'll make that very clear. Targeted therapy comes in a pill form. In veterinary medicine right now, what we have available all is in a pill form. So you get to give it at home for your pet and you're not having to bring them in and get IV chemo and so forth. This does not mean that there are no side effects. And I think a lot of times people think, oh, it's easy. There are no side effects. It only targets the mutation that's causing the cancer to grow or spread. No, it also can have side effects.
So with this dog that lived 9 months when normally 12 months is 0%, high quality of life.
Great quality of life. And I have no doubt that the Phytocure played a major role. I think a lot of times people say, well, how do you know it wasn't the cannabis? Or how do you know it wasn't? Well, I can tell you the dog stopped its herbs, I would say within a few months because it was getting some upset stomach. And so we had to weigh which ones we wanted to continue. And so if you ask like my inner gut feeling as a doctor, I would say I think it was probably some type of additive or synergistic effect that occurred between the cannabis and the targeted therapy. I see that in practice a reasonable amount of times, actually. And we have that one FDA-approved targeted therapy called Palladia. And so I've been using that now since it's come out for 12 years, 15 years, however long it's been out. And oftentimes, at least this is my experience, I have not publish anything to confirm this. I'm sure I have enough cases, to be honest. I just need to take the time to put it together and actually see the dogs that are on a combination of cannabis and targeted therapy. I seem to see really good responses, and I wonder if there is that synergistic action that's occurring. So that would be my gut feeling, but who knows? I can tell you that there was something that was different with this particular dog and any dog that I've treated with this disease ever in the past. And this is the only dog with this disease that I've seen that I put on Phytocure.
So do you basically recommend Phytocure for all of your cancer patients?
It depends on the cancer, right? So a dog that has this tonsillar squamous cell carcinoma that I know standard of care doesn't work well, why not give them a chance and say, I've tried it a ton of times with standard of care. It doesn't work. So let's try it.
But something like lymphoma that you would normally use the CHOP protocol, you wouldn't do it with.
No, absolutely not. I would use standard of care because I know that works better. So understanding which tumors tend to respond really well to standard of care, which ones don't, and then having the conversation with the owner, understanding it's not an FDA-approved drug, right? And so you have to understand that we're using it off-label, and let's go through the potential toxicities, which usually are GI. There's very few other ones, but there could be, and we just need to be a team, right, in this type of journey together and make sure that we're talking and making sure the pet is feeling good and things are going well, but really picking and choosing which cancers and which cases also might be a good fit, right? There's some cases where coming in and getting IV chemotherapy might be best for the cancer, but might not be best for the dog. And there's some dogs that are just so anxious and do not want to come into the hospital or are very aggressive and can't come in and get sedated every time for chemo. Those are the dogs that I think targeted therapy, if it's going to be Phytocure or Palladia that already exists, even chemo in a pill form could be a reasonable option. I think that's where personalized medicine is so important. And Phytocure is clearly looking at the genomics of the dog's tumor, not just the pet itself. And that's where I think when we combine genetic evaluation of the patient and genetic evaluation of the actual cancer, that's where I think magic will really, really happen.
Is this the future of veterinary and maybe human medicine?
Totally. Absolutely. And I think the combination, like once you can figure out the makeup, like if you think of almost like a map of you as an individual and a map of your actual tumor, you put that together, you almost like overlaid on top of each other and you say, well, what should I use? And then, of course, as an integrative oncologist, I say, why am I only looking at Western options, right? Or pharmaceutical options? Why can't I use things like turmeric or use Chinese herbs or cannabinoid therapy or something that also may be able to help? Once I get that map, I can find different ways of targeting things, right? It doesn't have to just be pharmaceutical.
Wow. I think we can all agree that this week's episode was jam-packed with great stuff about DNA, cancer-causing genetic mutations, and the science behind targeted cancer therapies. Ben Lewis gave us so much more than I was expecting from our interview, but I'm so glad he did because it helps to have a deeper understanding about the different treatment options available for our dogs, especially when those options are as revolutionary as this one. If you need more info about Phytocure, check out this week's show notes and the verbatim transcript on our website, dogcanceranswers.com, where we will share the links mentioned in this week's show, as well as provide Ben's contact information so that you can reach out to him directly to share your thoughts, questions, concerns, comments about Phytocure. If you like our show, please show us your support by liking us on Facebook and by subscribing to Dog Cancer Answers on YouTube or Spotify or Apple Podcasts or through your favorite podcast app. And if you want to become one of our ultimate channel supporters, make sure that you tell your friends, your family, and most of all, perhaps, your veterinarian and their staff about Dog Cancer Answers. Doing this will not only help our podcast continue to grow and reach more people, but it will help us to connect to other dog lovers and dog guardians just like you. Up next, we have a quick message from this week's sponsor because, well, that's how we pay the bills around here. Stay tuned, though, after the sponsor break. I'll be back to tell you how you can call us with your own dog cancer question and how that question could even turn up on a future episode of Dog Cancer Answers. We'll be right back. Did you hear those delightful touch tones? Well, Dog Cancer Answers has a 24-hour-a-day, 7-day-a-week recorded listener line, especially for you. If you have a dog cancer question or an idea for a future show, just call us and tell us about it. We check our messages often, and we'll make sure that your question is addressed by one of our veterinary experts, and it might even appear on a future episode of Dog Cancer Answers. The number to call is 808-868-3200. That is 808-868-3200. Remember, it is available all hours of the day, so you can call us anytime and leave your question on our recorded listener line. And if you're not ready to call, but you still have a question about dog cancer or a potential show topic, you can share that with us. Connect with us in writing by going to our website, dogcanceranswers.com, and clicking on that link that says, Ask Us a Question. We read all of our messages, and we look forward to hearing from you. Well, that does it for today. Again, thank you for tuning in. Until next time, I'm James Jacobson, and from all of us here at Dog Cancer Answers and Dog Podcast Network, we wish you and your dog a very warm aloha.
Thank you for listening to Dog Cancer Answers. If you'd like to connect, please visit our website at dogcanceranswers.com or call our listener line at 808-868-3200. And here's a friendly reminder that you probably already know. This podcast is provided for informational and educational purposes only. It's not meant to take the place of the advice you receive from your dog's veterinarian. Only veterinarians who examine your dog can give you veterinary advice or diagnose your dog's medical condition. Your reliance on the information you hear on this podcast is solely at your own risk. If your dog has a specific health problem, contact your veterinarian. Also, please keep in mind that veterinary information can change rapidly. Therefore, some information may be out of date. Dog Cancer Answers is a presentation of Maui Media in association with Dog Podcast Network.